Please.
Thank you for the introduction. I would like to thank Professor Valori, who actually helped me very much by first expressing the importance of genetic replication for life, and second by initiating the discussion on the definition of the life. So that I would say that probably everybody has his own definition of what is life. And I think that nobody would be satisfied by any other's definition of what is life, because everybody can say something his own on this topic, especially in this audience. But I would suggest, just for today, to assume that my definition, that life is a form of propagation of a genetic material. Of course, it's a very narrow definition, but just for this lecture. And at present, the simplest imaginable way for accomplishing this goal, that is propagation of genetic material, is provided by the concept of the RNA wall, because RNA is the only type of molecule that can serve both as templates and catalysts for their own amplification. And by today, a lot of data supporting this concept was obtained. First, that nucleotides can spontaneously form under conditions that existed on the early Earth or a similar planet. Activated nucleotides can spontaneously polymerize into long. I indicated here 40 nucleotides, but answer indicated that it can be up to 50 nucleotides long strand. RNA molecules can spontaneously recombine to produce even longer strands. And pools of random oligonucleotides, synthetic oligonucleotides, consisting of billions to trillions molecules, which actually is a very minute amount of RNA. It's only 100 to 10 micrograms of RNA. All this contains molecules from which one can select ribozymes, which with virtually any desired catalytic function. And selected ribozymes can catalyze the synthesis of RNA strands that are complementary to RNA templates. These are all very strong, solid evidence or support for the concept of RNA wall. However, there is at least one problem that yet to be solved. The synthesis of synthesized complementary strand and the template form a double helix along the entire length. And as a result, the template and the synthesized complementary copy are locked in the double helix and therefore unavailable as templates for the synthesis of more RNA copies. Hence, no propagation of genetic material or life becomes possible. Here we come to the paradox which was announced in the title of my lecture. According to Webster's dictionary, paradox is something inconsistent with common experience of having contradictory qualities. And there are contradictory qualities or inconsistent properties with complementarity, with double helix. Complementary nucleotides become complementary only in the double helix, because in the solution, any randomly chosen pair of nucleotides would be to some extent complementary, that is capable of formation hydrogen bonds. However, only in the structure of double helix they become complementary in the Watson and Crick's manner. So that T matches A and G matches C. However, therefore, to build a complementary strand, one must have a double helix. However, to enable further replication, the template of the complementary strand must remain single strand, just out of double helix. once again, double helix is needed for a template-directed RNA synthesis, but it prevents RNA amplification. And like the artificial ribozymes, hypothetical replicase the ancient RNA vault must have encountered with this problem, as was stressed by Chetverin. In a polymerase chain reaction, the paradox is overcome by temperature cycle. If we start with double-stranded DNA, then we raise the temperature to melt it into single strands, and then each of the two complementary strands is copied by a polymerase to make their own double strands. However, temperature cycle is not a proper solution for the conformation paradox in the RNA vault, as it generates another chemical, in this case paradox. Different cations, magnesium and calcium, are needed for the catalysis of RNA synthesis at low temperature. However, but they catalyze RNA hydrolysis or depolymerization at the high temperatures required for melting DNA doublets. And now I come to replication of RNA phages. In late 60s, RNA phages were associated with a breakthrough in molecular biology because many discoveries in both translation and replication was made using RNA phages, such as MS2 or 2-beta phage. And one of the classics of studying RNA replication with case of 2-beta phage, 2-beta replicase was Chetverin. And here is his figure from a lecture on Oparin-2014 Symposium on quantitative biology. And when Watson and Crick proposed a hypothesis model for double helix, they started from the view that double helix is needed for amplification of genetic material. So that, and they postulate that every amplification or replication of genetic material must go through formation, intermediately, at least formation of double helix. And so did Chetverin. He studied intermediates of replication reaction with 2-beta RNA and found that firstly, RNA-resistant material is formed, which they interpreted as formation of DNA, of DNA-like double helix, RNA double helix, and only then single-stranded RNA appear. So they postulated the first step of RNA replication is the synthesis of double helix, double strand, and then this double helix was read by 2-beta replicase by either displacing one of the strands from the helix or by initiation at different point of the double helix. However, further studies have shown that there are no double-stranded intermediates in 2-beta RNA replication. This was mainly did by another class of 2-beta studies, Charles Spiegelman, Weiss, sorry. If we follow Spiegelman, observe intermediates or products of RNA replication by phenol extraction of the reaction mixture and then analyzing the mixture by gel electrophoresis, then we can see that first we observe formation of double-stranded RNA and only then formation of single-stranded RNA in agreement with the observation of Spiegelman. However, what did Weiss? He first subjected the sample to RNase degradation prior to phenol extraction with RNase that is single strand specific, so that it would degrade single stranded RNA it would not degrade double-stranded. So that if some material remains after this treatment, it was indeed double-stranded. And all single-stranded RNA will disappear. And it appears that actually all material from the reaction is sensitive to single-strand specific ribonuclease. It is no double-stranded DNA is actually formed. And Weissman concluded that double-stranded RNA in this case is artifact caused by phenol extraction of the analyzed sample. And he also concluded that like ribozymes, the replicase cannot use the double helix as a template. Nevertheless, it somehow, the replicase somehow solves the conformation paradox, so that it managed to build a complementary strand, yet keep the template and the growing complementary strand single strand. How does the replicase overcome the conformation paradox? Weissman suggested two possible solutions to this problem. First one is the double helix is unwound by qubit replicase itself, acting like a zipper, to separate the template and the complementary nested strand, which are then stabilized in the single-stranded conformation by the intermolecular secondary structure. However, the double helix formed by complementary RNA strands are thermodynamically more stable than are intra-strand secondary structure. This is supported by the observation that if a mixture of complementary strands is annealed, that is melted and then slowly cooled, that the strands are completely converted into double helix. Then, within the replicative complex, the template and the nested complementary strand are close to each other, so that which favors their annealing. Then, and finally, the strands immediately collapse, just experiments performed by Weissman, into a double helix under action of proteases and beta-gas that cannot affect the stability of the RNA segment structure, but destroy or unfold the protein structure, so that secondary structure of single strands cannot keep single-strandedness of the replicated information. Possible solution number two. The unzipped strands are kept from annealing by a single-stranded binding protein that holds the strands along the entire length. However, the replicative complex observed in qubit replicase reaction remains single-stranded even in a purified self-resistant that contains no proteins but qubit replicate. And possible solution number three, Weissman hypothesized that replicase may hold the 3' end of the template and the 5' end of the nested strand during the entire replication cycle, forming like a butterfly structure. This poses topological constraints to binding the strands into the double helix. However, there was observation that several nested strands can simultaneously be synthesized on the same template strand. In this case, the same concept could work if the 5' and 3' termini of the template are held together, not by elongated replicase, but something else. And if we go back to the late 60s, then there was a paper by Spiegelman who observed the template activity of the genomic RNA of H2 beta drastically drops according to this fragmentation into two halves. It looked like beta replicase may sense during initiation if the template strand is intact or not. And to explain this observation, Spiegelman suggested the Afro model, that the template strand could form a circle if it had complementary termini capable of base pairing. The replicase could then recognize the terminal helix, or panhandle as it's called today. At that time, there was no information about sequences of termini of the beta RNA. Later, it occurred that indeed, replicable RNAs, including 2 beta RNA, indeed have complementary termini. So that every RNA has the oligo C on the 3' end and oligo G on the 5' end. However, the complementary stretches are too short, just 3 to 4 nucleotides, to form a stable circular structure of the template. And Weissman pursued this hypothesis by suggesting that inability of fragmented template to replicate might be a mere consequence of the fact that the initiator 3' end of the synthesized complementary strands cannot be synthesized. And so that complementary strand cannot serve as a template, so that would preclude exponential amplification of the template. So that this simple explanation of the fact observed by Spiegelman just proved the hypothesis of Spiegelman. And about 40 years later, we decided to reinvestigate this point. We started this issue an example of our small replication RNA. This is RQ-135, which means RQ means replicable by 2 beta replicase, and 135 means the length of this RNA in nucleotides. And we also investigated other small replicable RNAs, and all known replicable RNAs were found to be capable of formation of a hairpin that involves the 3' and 5' terminal structures. In case of this particular RQ-135 RNA, the existence of these hairpins was supported by data ribonuclease treatment. This, if we do it with single strand, specific non-beam nuclease RNAs, then these positions that correspond to the proposed loop are attacked. If we treat with RNAs DID, which is double strand RNA specific, that all this sequence is attacked. So that this observation supports the existence of such a terminal helix. And then we decided to investigate if this formation of such a helix that implies that interaction of the 5' and 3' end is important for and has any functional importance. To this end, we mutated, damaged the RNA structure in different ways. First of all, we clipped the RNA into about halves, and also introduced point mutations at the 5' end, and then investigated if this would somehow affect the ability of this RNA to be replicated by 2 beta replicase. And I would remind you that replication begins with the 3' end of RNA. We observed very drastic changes of the ability to replicate after these changes. When we split the RNA into 2 halves, the 3' fragment from which replication begins began very inefficient replay. The efficiency falls about 30-fold, 30 times. And then, when we admitted back the 5' and 3' fragment, so that this almost reconstituted the original 135 sequence, but with a cleavage inside, then it reconstituted much of the activity of the template, but not fully. And even point mutations introduced in the 5' end appear to decrease the template activity of this RNA. Especially, drastic effect was observed when this mutation was introduced in the 5' fragment and new to the 3' fragment. Okay. The point mutation in the 5' end increased the requirement of the RNA application for the concentration of the initiator nucleotide, which is GTP. It is seen that when point mutations are introduced in the 5' end, the replication which initiates with the 3' end becomes more dependent and requires for high concentration of the GTP. Finally, and also mutations in the 5' end decrease the rate and yield of initiation of the RNA. To observe initiation, we incubated the template incubator replicase and GTP only for different time. And then I added auric-carboxylic acid, which is an inhibitor of reinitiation and allowed elongation by addition of all missing nucleotides. Then we found that if a template had a mutation in the 5' end, that the rate and yield of initiation was about 2 times lower than wild-type template. And finally, mutation in the 5' end of the template destabilized the post-initiation replicative count. In this case, we again incubated the template with replicase and GTP by for a constant 10 minus 9. Then added auric-carboxylic acid and incubated with different time with this inhibitor of reinitiation and observed the rate how this initiation complex is composed. By addition at specified time, missing nucleotides and observe the formation of full-size complementary copy. And it appeared that even point mutations at the 5' end drastically decrease the stability of the replicative complex, so that the 5' end of the template interacts with the 3' end and at the initiation step and thereafter. And we hypothesize the terminal helix of the template might, by itself or with the assistance of replicase molecule, fasten the template in the circle conformation and thereby help to keep the replicative complex single-stranded during the entire elongation fight. And similarly, the conformation paradox might be overcome at RNA replication in the ancient RNA form. There is a growing body of evidence that viral RNAs and even eukaryotic mRNAs today form circles. This feature might be a relic inherited by the contemporary DNA world from the RNA world, in which a circular structure was a prerequisite for the ability of the genetic material to propagate. Thank you very much.